Hello again. In the previous section we started with lysosomes, we completed lysosomes. Now in continuation with that, when we use the word lysosome, normally we are talking to the structures which are present in animal cells. So you can also consider these as animal lysosomes. Lysosomes are present in both plants as well as in animals. So whenever we are using the term lysosome, normally we are referring to the lysosomes which are present in animal cells. Then what are plant lysosomes known as? They are known as spherosomes. So spherosomes are plant lysosomes. So lysosome term is common, but normally whenever it is used independently, the thing that comes to our mind is we are talking about animal lysosomes, and spherosomes exclusively term given for plant lysosomes. Let us quickly compare these structures. We have seen the structure of lysosome in detail. These are spherical bodies. Same is the case with spherosome, though smaller in size. Lysosomes were discovered by Christian D. Dewey. This also we did in the previous section. And spherosomes were discovered by Perner. Lysosomes are polymorphic. Polymorphic means they exist in 4 different forms: the primary lysosomes, secondary, tertiary, and the autophagosomes. In this case, in case of spherosomes, there are no forms, but depending upon what is stored inside, there are types. So these are not forms. Now what are the types of spherosomes? It is decided by what is present inside. When we use the term spherosome, they have the hydrolytic enzymes same as that of the animal lysosomes. So they have hydrolytic enzymes. But sometimes plant lysosomes, apart from the enzymes, also have some stored material or substance like alluron. So alluron is a protein. So then in that case they are known as alluron grains or particles. Here it is alluron proteins which are stored. So it could be a storage thing also. Other than proteins, it can also store lipids. Then they are known as simple vesicles, or sometimes also known as vacuoles. But this vacuole should not be confused with the regular plant vacuole which we have. So here there are types depending upon what is present inside the spherosome. Whereas in case of this, we saw forms, and these forms were on the basis of how the matrix changes, whether it is inactive, active, or what exactly it is doing. We talked about 2 main types or 2 main categories, in the sense that primary, secondary, and tertiary lysosomes were responsible for digesting the foreign bodies or foreign food item. They are known as heterophagosomes. And the 4th type, which was digesting its own old organelle, was called autophagosome. So heterophagosome was digesting outer food material. And as we are talking about animal lysosomes, we know animals are heterotrophs. So they are obtaining their food from some other plant or animal, and they have to digest it. In plants, we don't have this condition because they synthesize their own food. So hetero and autophagosome condition is not there. It is only the reserve food material which they have needs to be digested, and that is done with the help of the spherosome. We will come to that in a minute again. Certain lysosomes, they behave like heterophagosomes, the first three categories, and fourth category, that is autophagosome, digests its own. If a lysosome performs both the functions, then it is known as ampilysosome or amphilysosome. If it performs both the functions. In case of this, only spherosomes are responsible for digestion of substances because they have hydrolytic enzyme. Areas or cells where lysosomes are present are the cells where more digestion or breakdown of substances is required. So we took the example of WBCs, which are going to engulf the foreign particle and digest them, plus osteoclasts. These cells also destroy the old and worn out bone cells, that is osteocytes. In case of this, the spherosomes are found in the areas where food is to be digested. So what are those areas? Endosperm, for example. So endosperm, which is the reserve food material, if that is to be digested, that will be done with the help of spherosome. Plus they are also found in cotyledons, where again reserve food material is present which needs to be digested, and then this digested food will be supplied to the developing embryo or the baby plant. Aileron structures are stored in the seeds or fruits where this aileron protein needs to be stored. Say for example in maize, the outer part of the maize grain has aileron grains. Vesicles or vacuoles again would be present in the storage areas where the lipids or maybe oils are stored. So depending upon where which particular substance is stored, aileron grains and vesicles would be present there. Spherosomes would be there in case of where digestion has to take place. So because of this hydrolytic enzyme, two places where reserve food material is stored, that is endosperm and cotyledon, we find maximum spherosomes. In case of lysosomes, again there are certain metabolic disorders which have been reported. For example, lysosomes are supposed to remove all the foreign particles, or they need to digest the things. In case of lysosome, one metabolic disorder which has been reported is known as Pompe's disease. Pompe's disease basically is due to accumulation of glycogen. Normally this glycogen should be broken down by lysosomes, but if the enzyme required for breakdown of this glycogen is absent, then this glycogen will be accumulated in the lysosomes, and that results into this metabolic disorder which is called Pompe's disease. One more thing which is reported about lysosome is that these hydrolytic enzymes which are present in lysosomes can cause mutation and can result into cancer. So they have also been considered responsible for cancer. Not directly, but because these enzymes can cause mutation. In case of spherosomes and all these structures, we don't have such distinguished disorders or metabolic disorders. I'm sure there are few changes which must be taking place if these structures do not work properly, but we don't have a well-defined disease named so far. So this is a simple comparison between the animal lysosome and the plant lysosome, which we call the spherosome. Now let us take the endomembrane system. We have been talking about this endomembrane system in almost all the organelles so far. So let us see which all structures make this endomembrane system. The endomembrane system, it comprises of the plasma membrane and organelle which are with membranes. Which all organelle or structures are part of this? Plasma membrane is a part, endoplasmic reticulum, both RER and smooth, that is SER, both are the parts of this. Then Golgi complex or Golgi body, even lysosomes. And how do they internally change into from one structure to the next structure? Let us take a simple diagram to understand what exactly is happening in this. This is the plasma membrane. When the cell undergoes endocytosis, in this plasma membrane we see a cup-like invagination formed, and in this cup would be taken the food particle. When the food particle moves in, this cup-like structure will change into a food vacuole. So this is a food vacuole. Now depending upon solid or liquid, you can call it phagosome or pinosome. So right now we are just calling it phagosome, assuming that it has the solid food inside it. Now this food which is inside needs to be digested. How would this be digested? It would be digested by lysosome. How is this lysosome formed? The endoplasmic reticulum, on the outer side it is in the form of tubules which are wider and without ribosomes. As we move towards the nucleus, we get these cisternae, and these cisternae are interconnected. Coming closer to the nucleus, we have the Golgi complex. So here is the Golgi body. Now what is happening here is, on these endoplasmic reticulum are ribosomes. What are ribosomes doing? Ribosomes are helping in protein synthesis, and we know that endoplasmic reticulum helps in transport of these proteins. So these proteins get transported through this, and here they are packaged in vesicles. So these vesicles which are getting pinched off from endoplasmic reticulum, these vesicles are with proteins, so they contain the protein. So they get transported from ER, they will go to Golgi body. They will come closer to the Golgi body from its convex side, that is cis or formative, and it will join the Golgi body membrane. That means the protein which is present in this vesicle is now transported to Golgi body. In Golgi body, it will undergo modifications, transformation from primary protein to secondary to tertiary, depending upon what is required at the end of it. Say enzymes are required for digestion, so these proteins which are synthesized, they are primary proteins, they will be converted into tertiary proteins, they will get into that three-dimensional structure. All this will take place in Golgi body if it is to be used as an enzyme. If this protein is to be converted into something else, those changes will also take place in Golgi body, like glycosylation. Glycosylation means addition of oligosaccharides to the protein molecules. Now all those transformational changes take place here, and the protein is converted into enzymes now. So here in these vesicles was simple protein which was not acting as enzyme. Now these vesicles have enzymes. Can we call these vesicles as lysosomes now? So the vesicles with enzymes which are pinched off from Golgi body, they have inactive enzymes, so they are lysosomes. Which type? The primary lysosome. Now this primary lysosome with its enzyme fuses with this particular food vacuole. So next stage is this lysosome and this food vacuole, they fuse. So this is the part of the phagosome and this is the part of the primary lysosome. Now because it has fused, this has changed into the secondary lysosome. Digestion will start because of these enzymes which are present here. After digestion, now what remains inside is only undigested food. What needs to be done with this undigested food? It should be thrown out of the cell. So this structure will again come closer to the plasma membrane and will open here, and this undigested food will be thrown out. So the vesicle or the structure which is having the undigested food is the tertiary lysosome. And this is ephagy, the removal of undigested. So in this entire thing, all the membrane bound structures participated. This much part of the membrane, plasma membrane, pinched in, and the same size plasma membrane again went back to plasma membrane. So if this much part was removed, the same part was added after some time. And during this entire process, see, ER also participated, Golgi also participated, all primary, secondary, tertiary lysosomes, and ultimately it came back to plasma membrane. So when we are saying plasma membrane, ER, Golgi and lysosomes make the part of endomembrane system, this is what is the actual process that we are talking of. Hi, this is Dr. Neela Bakore, and today the structure which we are going to take up is ribosome. When we talk of organelle term, organelle are membrane bound. Ribosome is a structure which is not a membrane bound structure. It is only a protein and RNA particle but without membrane. So let us take ribosome. The discovery or historical part first. It was observed by a scientist named Claude. Its structure, the ultrastructure which we will be studying today, that was given by two scientists, Pallade and Cuff. The term ribosome was given by same scientist, that is Pallade, and that is why ribosomes are also known as Pallade particles. Pallade's particles. Because of the same reason that he gave the term as well as the structure. In plants, ribosomes were first observed by two scientists, Robinson and Brown. And the plant in which they observed these ribosome was Vicia faba. So this was the plant where the ribosomes were first observed by these two scientists. Where are these ribosomes present or found? They are found in all cells. That means they are found in prokaryotes or prokaryotic and eukaryotic cells. There are exceptions again in eukaryotic, like the mature mammalian RBC. We don't have ribosomes there. But prokaryotes and eukaryotes, all the cells, they have ribosomes. Ribosomes can be freely scattered in cytoplasm, as we have seen in prokaryotes. So they can be free in cytoplasm, which we see in all prokaryotes, and in eukaryotes they can be free as well as bound. So free or bound in eukaryotes. Free means again in cytoplasm, and bound means attached to the membrane of ER or vesicles, which we have seen when we were talking of endoplasmic reticulum. So free or bound, that is seen in case of eukaryotes. The free ribosomes which we are talking about, they are found in the cytoplasm. But two other organelles, that is mitochondria and plastids, also have ribosomes. So the ribosomes which are present in mitochondria and plastids, they are known as organellar ribosomes. Organellar ribosomes. The ones which are found in mitochondria are called mitoribosomes, and the one which are found in plastids are known as plastidoribosomes. Ribosomes are of two types on the basis of their sedimentation coefficient, 70S type and 80S type. S, which we are writing here, is actually, I am writing this S here, S stands for sedimentation coefficient, or Swedberg's unit. Swedberg's unit. It basically tells at what rate does it sediment. So there are two types, 70S and 80S. Now let us see the differences between these two. 70S are found in prokaryotes and eukaryotic organelle, that is mitochondria and plastids. So when we are talking about mitoribosomes and plastidoribosomes, these are actually 70S type. 80S are found in eukaryotes, that is the ones which are in the cytoplasm free, as well as the bound one which are attached to the endoplasmic reticulum. Each ribosome is made up of two subunits. Normally when we draw ribosome, we make a larger subunit, and attached to that larger subunit is a smaller subunit. So here also there are two subunits, in 80 also there are two subunits. The two subunits are 50S and 30S. Don't get confused with the addition number. 50 plus 80 comes to 50 plus 30 comes to 80. But this is not a combination thing. It is not a number thing. It is the sedimentation rate, the rate at which it would sediment. So one is 50S, which is the larger subunit, and 30S, which is the smaller subunit. In case of 80S, again there are two subunits. The larger subunit is 60S and the smaller subunit is 40S. Again, let us not get confused with the addition thing. This total sedimentation coefficient is not the addition number of these two. These three things are different. As a whole, it would sediment at a rate of 80S, and individually they would sediment at a different rate. So two subunits here. This is the larger subunit. This is the smaller one. In this case, 60S is larger and 40S is the smaller subunit. Normally, these subunits remain separated. They would join only at the time of protein synthesis. These 70S ribosomes have proteins and rRNA. The ratio of proteins to rRNA is 60 to 40. It is also written as 1.5 is to 1 ratio. Whereas in case of eukaryotes, the protein to RNA, rRNA ratio is 50-50. So it can be written as 1:1 ratio. So the ratio is different, but structurally, functionally, they are pretty much same. These are two types. We'll add a couple of points little later. Let us see the structure of ribosome first. The larger unit or larger subunit has a projection on the upper end, which is known as protuberance, a ridge and a body. So this part is a protuberance. Protuberance is nothing, just a bulge, and this is a ridge, and the remaining part is the body. This is the larger subunit. The smaller subunit has a head-like thing. Then here there is a depression, which is known as a crest, and body. So there is one head, a crest, and body. The larger subunit attaches to the wall of endoplasmic reticulum with a protein called ribofourin. We talked about ribofourin and endoplasmic reticulum. Two ribofourins, that is ribofourin 1 and 2, it's a protein, and larger subunit attaches to ER. Important, very, very important thing. Smaller subunit has a site for attachment of mRNA. Ribosomes are going to help us in protein synthesis. Protein synthesis means the message is going to come from DNA, and then ribosomes are going to help in translation of those codes from mRNA to protein synthesis. So it has a site for attachment of mRNA, that is messenger RNA. So larger subunit attaches to endoplasmic reticulum, and the smaller subunit has mRNA attachment site. Larger subunit has 3 sites. Now we will draw those 3 sites in slightly different manner. 3 sites. These sites are known as A site, P site, and E site. A site is for amino acyl. It is called amino acyl site, where the amino acid with tRNA is going to come and get attached. P is known as peptidal site. This is the place where the peptide bond will be synthesized, and E is the exit site from where the tRNA is going to leave the complete assembly. This larger subunit also has one very important enzyme, that is a protein specially, and that enzyme is going to help in peptide bond synthesis. It is known as peptide transferase. So, this particular larger subunit has an enzyme called peptide transferase. Smaller and larger subunit. So, the enzyme predominantly found in larger subunit is peptide transferase. These two subunits normally remain separate. They would assemble only when protein is to be synthesized, and then the actual ribosome will be formed. Right now they are in the form of two subunits. Then they attach and form the ribosome, the complete structure. Let us see how these ribosomes are formed and what conditions are required for their assembly.